Oh, there we go. So the outline of the talk is that I start first with explaining why you would want to do single molecule, dynamic single molecule research, and then I will introduce to you the technology that LUMICKS has developed. The instrument that I will talk about today is called the C-Trap. I'll show you the experimental setup and I explain the technology, and then I will go over a few applications of the technology in looking at DNA protein interactions, specifically looking at the examples, looking at DNA editing, DNA repair, replication, and viral packaging. But why dynamic single molecule? We know that biological processes are highly dynamic and they're highly complex. Here is the famous animation showing the process of DNA replication. And as you can see, it's a highly dynamic and complex process. And we already know a lot about these biological processes, either from structural studies or from bulk biochemistry assays. However, the structural studies are, the data we get from those studies are static, but we do get dynamic information from the functional assays from biochemistry. But the problem is that those information we get, the averages over all the participants in a biological process, all the contributions. So looking at these biological processes at a single molecule level, you will be able to look at individual molecules and get detailed information about molecular mechanisms of these processes, which is not really possible using traditional techniques. So you can, for example, get more detailed information about the subpopulations of the molecules that are involved, or look at rare events or transients or short-lived events. So you would get way more information from these single molecule techniques, which, as I mentioned, it basically completes what we already know from these from structural assays and biochemical assays. Having said that, I will I will introduce to you the technology that LUMICKS has developed, but before that I start telling you about optical tweezers. So the technology that we're going to talk about is basically a combination of optical tweezers technique with fluorescence microscopy. But before getting into that, I briefly go over how optical tweezers work. So basically you would use a laser light to trap small objects. So in this case, you can see, for example, there's a polystyrene bead that it gets in the way of a laser beam. The beam enters and exits the bead, and as you can see, there's having this bead in the way of the beam would change direction, and that would cause a change in momentum of the light. But because of conservation of momentum, at the same time, the bead would feel a change in momentum as well. So if assume you have a highly focused laser beam and have a polystyrene bead, in this case, close to in proximity of that highly focused beam, the bead would feel a net force towards the center of the beam, as you can see in this diagram here. And basically, with using this technique, you will be able to trap or hold on to small objects or any biological sample of interest, in this case, that we're going to talk about today. So optical tweezers they can work as optical spring. So imagine you have a bead trapped in the at the center of the focused laser beam. If the bead would get displaced from the center, it would feel a restoring force that would bring it back to the center of the beam. So by monitoring the position of the bead, you can actually calculate the stiffness of this optical spring, and also measure the forces that are applied on the bead or any other object that is trapped. So this allows you to use optical tweezers as a highly sensitive force detector, which is how the application of optical tweezers in biology started at the first place. Which was for looking at forces that are basically it was used to measure forces in biological processes. And we know there's a range of mechanical forces in biological processes, ranging from femto to micronewtons. And having said that, it's optical tweezers is not only useful for measuring those forces, but at the same time, it allows you to trigger a specific biological process that you're looking at, or manipulate a biological sample that you're interested in, as I will explain later on how that manipulation is possible. But having said that, I will first introduce you to the technology that LUMICKS has developed, and the instrument is called the C-trap, which, as I mentioned, is a combination of optical tweezers with fluorescence microscopy. So the optical traps in the system, it could be a dual trap, which you would have two focused IR laser beams, which allows you to hold on to, for example, two polystyrene beads, as I mentioned, and attach it to a biological sample of interest, and either manipulate the sample or measure forces. That would be called a dual trap. Or you can have 4 of those traps, which is called we call it Q trap or quadruple traps. So it comes in both configuration. It is the unique thing about the system is that it allows for very high resolution measurements of forces and also displacements, and it's highly stable. As I mentioned, it comes with fluorescence imaging capabilities, which we have 3-color confocal scanning microscopy, which you will be able to to detect single photons. So it's highly sensitive, and you can also add super resolution STED microscopy to that. It also comes with a microfluidic system that I will show you later on, which allows for doing even more complex experiments. And the other unique thing about the system is the software. Actually, that it's called Blue Lake. You can fully automate your experimental procedure, and also it has a very easy to use software. As I mentioned, the instrument comes with the microfluidic system. Here is a picture of how it looks, and here also is a picture of a regular flow cell that we use in the kind of applications that I'll show you later. So these kind of experiments, we use laminar flow to have separate channels, to have different, for example, different proteins or different biological samples in each channels, and there's no, the channels are, there's no mixing between the channels, but at the same time, there's no physical boundary between them. So you can move the flow cell or move the stage and uh, take the trap or whatever biological samples that you have trapped with the lasers. You can move them easily between the channels. I think it would be more clear once I show you a few examples of the experiments and also the workflow in this video. So here's a short video showing you the system and a typical workflow. So that is where the sample goes in, sample holder, and it shows the microfluidic system. I already showed you how a typical flow cell looks like. Um, so as I mentioned, you can have different channels, different flowing and not mixing, and have, in this case, for example, you have bead in the first channel. You shine the two focus lasers into the bead channel. You trap two polystyrene beads, which are functionalized, for example, with estradiol. Then you move to the DNA channel, where you would basically fish for DNA. You wait until a piece of DNA is attached to two beads, and the DNA is also functionalized at the ends with biotin. And by moving the traps away from each other, you can apply force and make sure you have one DNA attached. Then you move to a different channel, which has the protein of interest, in this case, fluorescently labeled. And then you move to another channel with another protein of interest that is labeled. And then you can move into a buffer channel where you don't have any fluorescence, and you can start imaging using confocal scanning. So as you can see here, it allows you to, at the same time, you can, not only you can manipulate the DNA molecule that are attached to the beads, but at the same time, you can look at the proteins of interest that are bound to the DNA and get dynamic information from it, which I will explain later as well. And this is just showing that at the same time that you image these fluorescently labeled proteins that are bound to DNA. At the same time, you can do force measurements as well by moving the traps away from each other and applying tension to DNA. If you want to know more, you can always go to our website and look at the brochures or other videos on the website. So as I mentioned, the key thing here is correlation between force spectroscopy and fluorescence imaging, where at the same time that you're doing the fluorescent imaging, you can also either measure forces that are involved in a specific experiment that you're interested in, or as I mentioned, you can manipulate and trigger these biological reactions by applying force or changing the tension on the DNA that is attached to the bead. Here on the right, you will see it shows that you can basically do a 2D scan, getting a confocal scan of the whole complex of the beads and DNA and the fluorescent proteins that are attached to the DNA, or you can do a 1D scan, or as we call it, chymograph, which gives you information about not only what is bound to DNA, where it's bound, but also over time. It would give you more dynamic information about those bound proteins for DNA. So today I will go over a few applications of this technology for looking at DNA protein interactions, but of course there is more that you can do. You can look at the protein folding or unfolding, for example, or looking at the motor proteins acting on microtubules or actin. Before I get into the applications, I just want to let you guys know that if you're interested in the other applications of the technology, my colleagues are giving 2 other webinars in upcoming weeks that you can find the information on the website. One is coming up next week and on cytoskeleton dynamics, and another one on protein folding, unfolding following week. In addition to that, we also have started the Wednesday webinar series with all the conferences being canceled due to the current situation. It would be good to keep researchers up to date with the research that's going on in the field of single molecule dynamics. Unfortunately, the first two are already past, the first two webinars, but there is one more coming up next week that if you're interested, you can attend. But if you are interested in the ones that are passed already from the previous weeks, you can send us an email and we can send you the recording of those webinars. And we're also planning more Wednesday webinars in the following weeks. And again, you can find the information on our website or newsletters. So with that, I will start showing you a few examples of how you can use this force spectroscopy technique combined with fluorescent microscopy to look at DNA-protein interactions. So here again, it shows two beads trapped and a DNA is attached between them, and it basically shows you that you can choose any protein of interest. The proteins are involved in DNA repair, DNA organization, replication, any biological process that you're interested in. And if you label those, fluorescently label those proteins, you can in addition to force measurements, you can also get a lot of dynamic information. As you can see in this video here, it shows a protein that is involved in DNA repair. It's fluorescently labeled. It shows you how, where it binds. It binds all of on different spots on the DNA. And by doing confocal scanning, you can get you can basically look at these complexes moving along DNA. So kind of information that you can get from these kind of experiments are, as I mentioned, you can see where these proteins bind on DNA. In this case, it shows a snapshot of the video that I showed you earlier, and you can get and on the right here, you can see that how you can get the binding sequence. Basically, you see where on DNA, which sequence the fluorescent protein is bound to with a high precision. And also, as I mentioned, you can get these kind of chymographs or 1D scans, which allows for looking at dynamics of these fluorescently labeled proteins that are bound to DNA. So in this case, you can, for example, look at the binding and unbinding of these proteins. Looking at the chymographs, you can see a specific spot, how long it stays on the DNA, where it unbinds and then binds again. So you can get the K-offs and K-ons, or just binding kinetics information from these kind of assays. And here on the right, it also shows you, as I mentioned, you can detect single molecules with these instrument, and here is basically showing that you can follow a specific spot over time and look at the bleaching steps, which also tells you what is the size of that protein complex on that specific spot. So the first example I'm going to show you today using this technology is to look at the off-target binding of Cas9 complex. As you know, CRISPR-Cas9 is very popular these days, and it's used in gene editing. But in order to use it in a reliable way. You want to get the details about the off-target binding site, which could be a problem for gene editing. So in this case, it's a work done in lab at, Imperial College of London, where they use the C-trap to look at fluorescently labeled Cas9 complex. So in this case, you'll see again two beads are trapped, and there's lambda DNA attached between the beads. Again, as I mentioned, we use mostly streptavidin biotin interaction to attach the DNA to the bead. And the Cas9 complex is fluorescently labeled. And as you can see in this movie, you're going to start imaging the DNA and the complex, and then start looking at where this fluorescently labeled complex bind to DNA. And the interesting thing that they observed in these experiments was that at the beginning you don't see any binding to DNA, but as you move the beads away from each other, meaning that you're applying tension on DNA, you would start to see more and more of the fluorescently labeled complex bound to DNA. And in order to look at that more closely, they looked at this binding and unbinding of this complex under different forces. Here you can see again chemographs of the complex, showing here at the beginning, starting at lower force 5 piconewtons, where you can see only one spot on DNA, but as you increase the force going to 20 piconewtons, you start to see more and more binding of the complex to the DNA. And what they did here basically was that they went to higher and higher forces. Sorry, I think there's maybe people are asking questions. Yeah, so they went to higher forces, and they basically wanted to show that this binding and unbinding it's reversible. As you can see, when you go by, you increase the force and then you go back to lower forces, and you will end up with one spot again on the DNA. And when you go to higher forces again, you start to see more off-target binding. So this was a very interesting observation, which hasn't been looked at before, basically showing how these off-target bindings are triggered by applying tension on DNA. And as I mentioned, you can get the information about where these proteins bind, what sequences on the DNA they bind. So they got these histograms of the spots on DNA, and they basically showed that these spots are they're not random, they happen in a reproducible way. So if you want to know more about these studies, you can check out the paper. Another recent application of the system was to look at the virus-like particles and their assembly in vitro. Here again, the same kind of assay. You see two beads trapped, the lambda DNA is attached between the beads, and these virus-like particles are fluorescently labeled. And they, by doing a dipping experiment, meaning that having the DNA in the protein channel, keeping it for a certain amount of time and then imaging it, they started to see that these virus-like particles, they bind to DNA, and they also compact the DNA. As you can see in these snapshots, the DNA gets shorter and shorter, and at the same time you see accumulation of these fluorescently labeled particles on DNA. So as I showed you earlier, by looking at bleaching steps, you can estimate how many of these fluorescently protein particles are bound to DNA, and that's what the histograms down here show, that at different concentrations of the virus-like particles, what is the average size of the spots that they would see on DNA. And you can get, as I mentioned, you can get these information by looking at bleaching steps. So the other thing that I wanted to show you today is how you can use the force spectroscopy, or the applying force on the DNA to look at DNA mechanics. There's been a lot of pioneering work done on overstretching lambda DNA. So here it shows again the same 2-bit assay with a lambda DNA attached. And based on those studies that I mentioned, we know that as you start to apply higher and higher forces, you get to, on a double-strand DNA, you get to this overstretching regime, where you go from having double-strand DNA to single-strand DNA, which is called DNA melting. So what the movie here shows you is that by applying, moving the beads away from each other and applying higher and higher forces, you can actually see this DNA melting happening in real time. Here, the DNA is labeled with Cytox, a DNA dye that binds to double-strand DNA. So at the beginning, you see less of Cytox bound because it binds better to stretch when DNA is under tension. So first you start to see higher intensity of Cytox, but as you increase the force, go to higher and higher forces, you start to see these gaps on DNA, which shows that double-strand DNA is transforming into single-strand DNA, and Cytox doesn't bind to single-strand DNA. So you can either look at the gaps, appearance of the gaps, to detect single-strand DNA, or if you have, in this case, a protein called RPA, the replication protein A, that binds to single-strand DNA has been used. It's fluorescently labeled. So where the gaps appears, you see binding of this single-strand binding protein to DNA. And here the bottom panel shows that if you have the first two, shows the DNA and beads when the flow is off. And in this case, if you have flow on, you can actually see the other strand labeled with fluorescent RPA. So what kind of experiments can you do with these kind of assays? Here is an example of looking at proofreading activity of DNA polymerase. It's known that if DNA is under tension at higher forces, around above 40 piconewtons, you can trigger exonuclease activity of DNA polymerase. And here is basically what has been looked at. Again, it shows a chymograph of a lambda DNA. No, sorry, not lambda, it's a 8.3 kb DNA substrate labeled with Cytox. And as you can see at the beginning, you see fluorescent intensity between the beads. It's kind of uniform. And as the DNA polymerase start to act on DNA and making single-strand DNA, you would start to see these gaps, which indicate of producing single-strand DNA. And as I mentioned, you can, at the same time, you can look at the end-to-end distance of the beads, showing the displacement, and at the same time measure forces, which in this case, for example, you will see this step, the activity, you will see these steps in the activity of the DNA polymerase, and you can basically see at each steps, how many nucleotides does it go. So these are, as I mentioned, you can use any enzyme of interest. These are just a few examples that I thought might be interesting to show today. And one more application that I wanted to touch upon. It's for sake of time, I don't go into details of this work, but it's a very interesting work done by a lab in Imperial College of London, where they used they used the technology to look at cohesion activity in tethering DNA molecules. So one thing I mentioned here is that as I told you before, you can have the C-trap comes in two different configurations. One is the dual trap, the other one Q trap. So I chose this example to show you how you can use the Q trap, for example, to look at DNA protein interactions. And that's what has been used here. So there, as you can see, there are 4 beads, and each pair has a DNA attached to it. And by incubating, so they attach DNA to both pairs of beads, and then they incubate it with cohesion complexes. And I should probably mention here, if you don't know about cohesion are from these SMC complexes, structural maintenance complexes, which are known to, they have a role in chromosome, keeping sister chromatids together, basically tethering them. So in this work, they've looked at how cohesion complexes can tether DNA molecules. So by incubating the 2 pieces of DNA attached to 4 beads in a protein channel, they start to see these knots or bridges between the 2 pieces of DNA. But the interesting thing that they observed, as you see in these movies here, were that these bridges, they are highly dynamic and they are very easy to move. So what they're doing here is that they move one of a pair of the traps in a horizontal direction in both. You can see it in 2 different movies in both directions, showing that these cohesion complexes, at the same time that they're making these bridges, they can easily slide on DNA, which is a pretty interesting observation. And the other interesting thing they observed was that these bridges are they're very persistent to force. So what they do here, what they show here in this video on the right, is that by pulling on the pulling one of the beads in Y direction, the bottom one, they're applying tension or force on the complex to see if they can break the bridge between the 2 pieces of DNA, but they only see that the attachment between the bead and the DNA breaks. And as you can see, the bridge or the knot stays even at going to higher forces. So based on these observations, they concluded that cohesion is capable of making these permanent intermolecular bridges, that they can slide very easily on DNA, but at the same time it resists higher forces, and they predict they have to go to 80 piconewtons or higher to break the attachment between the 2 pieces of DNA. So if you're interested in knowing more about this paper, I recommend you check it out. It's a very nice work. And at the end, I will just mention very quickly the application of the same technology, but without looking at fluorescence in DNA packaging. And in this case, DNA packaging by virus, bacteriophage. Here is just a cryo-EM reconstruction of the bacteriophage motor and its capsid. It basically compacts, it packages DNA into the capsid. And what they have done here in this assay is that they attach two beads to two ends of end of a DNA substrate, and the other end is attached to the capsid of the virus. And by looking at the displacement of the bead, they track the activity of the motor. And as you can see in the graph here, you can see the virus packages the DNA into the capsid in a stepwise manner. And if you zoom in into the different steps, you would see these pauses, these red regions, which they call dwelling times, and then there are these blue ones that it's basically burst of activity. So again, showing a stepwise packaging of the DNA, but by looking at these duration of these dwell times or burst times, and also how much DNA is packaged at each step, you can get more information about the how the motor actually works. And a lot of work has been done in Bustamante lab, and this is also an experiment that was done on a C-trap. But if you're interested in the paper, there are papers from Bustamante lab that you can look at, but this is basically reproducing some of those experiments using the C-trap. So I hope I was able to give you some insight and also some motivation to use this technology in looking at the dynamic single molecule. But I just wanted to, at the end, I wanted to mention that at LUMICKS, we don't just provide the system or the technology, we also try to help the researchers who are interested in doing dynamic single molecule research, and hopefully we help with breaking any barriers that there is in getting more people doing these kind of experiments. And how we do that is we try to help with optimizing the workflow by helping with experimental design, sample preparation, and training of how to do the experiments, data collection, data analysis, and so on. But recently, in order to help researchers even more, we've started making these biochemistry kits and services where allows researchers to, if they don't have the expertise in the biochemistry part, we would provide those services so that they can focus on the experiment. And the other thing that we're working on is launching this script sharing platform where people can share their scripts either for data analysis or automating the workflow of their experiments. And this would be helpful not only to C-trap users, but anyone who is doing these kind of dynamic single molecule experiments. So as I mentioned, you can find information about the different biochemistry kits and services that we provide on our website. And also you can find more information about the script sharing platform. It's not ready yet, but I think it would be ready by the end of May or early summer. But again, you can find information about it on our website or in our newsletters. With that, I would just remind you of the dream that we all share at LUMICKS is to, as I mentioned, make these kind of dynamic single molecule analysis accessible to a wider range of scientists. And we're hoping to be able to improve human health by unlocking single molecule and single cell analysis. And we are a growing team. The company is based in Amsterdam. I don't know if we mentioned that or not, but we have application scientists in US and in Europe, and we are a team of passionate scientists, and we're here to help whoever is interested in doing these kind of experiments. And with that, I will thank our collaborators all over the world that make it possible for us to show you all these cool data. We have over 50 systems all over the world. And with that, I also thank you for listening, and I'll take any questions that you might have. I think there are a few questions, but we'll go over them. Yeah,
thank you very much, Nasrin. So we have a few questions. So if you have any questions, just feel free to type it in the Q&A region. So the questions that you got so far is, so the first one is from Rico. So what is the range for length of the DNA that can be trapped between the beads?
Okay, so the range, the shortest that we've used is about 1 kb, which is about like 300, a bit over 300 nanometers. And the longest that I showed here is lambda, which is about the contour length is about 16 micron, but of course you can go to larger longer DNA substrate as well, because the range of the motion of the beads are larger than that.
Perfect, thank you. Rico, if you have any follow-up questions, just feel free to type it. So the other two questions comes from Rama. So the first one is, how do you differentiate between photobleaching and unbinding events in chemographs?
Yeah, that I think you need to do a lot of, you need to get the statistics. Basically, you need to look at the number of complexes bound, and if you do those kind of bleaching experiments that I showed earlier, you can kind of get an idea of what is the, like, how long does it take for the specific laser power, and like the imaging settings that you have, how long does it take for a spot to go away, basically, or bleach. So once you get those time scales, then you'll be able to, of course, if you see the spot disappearing in a shorter time, you would count it as unbinding. So I hope that would answer your question, but you need to get those bleaching time scales first, and you would get it by looking at multiple, basically look at the population of molecules.
Yeah, Rama, write us if you have any further questions. The second question that Rama had is that how do you find out the orientation of DNA molecule to assign the region where the protein binds?
Well, you can you can get the readings from the. I hope I got your question right, but for example, if you want, the way I understand is that how can you make sure that it's completely horizontal, right?
I think it's more along the lines, but right, just again.
So you can check the X and Y of the two.
I think it's more that, like, how do you see that your protein binds to this region of the DNA versus the other? So how do you know that it goes from 3' to 5'?
Oh, that you can do by designing your substrate. Okay, that's the, I thought they mean like the orientation of the DNA between the beads. Sorry, I got that wrong. That you can, in designing your substrate, you can choose, for example, like you would have a 3' end and you have a 5' end, like differently labeled. For example, one end of your substrate would be You can use biotin streptavidin interaction, the other, and another kind of like antibody, for example, DIG, anti-DIG. And or I know some people working with Lambda, sometimes they would put a fluorescently labeled spot, like they basically incorporate a sequence that is fluorescently labeled just to be able to tell the orientation of Lambda. So I hope that would answer your question.
Again, if you have follow-up questions, let us know if anything's unclear. So another question from Shiba. So are the microfluidic chambers provided commercially or can they be prepared in lab for multiple trapping experiments?
Well, we do provide the flow cells. Yes, it's commercially available, and you can have different designs of like whatever, depending on your assays. But you can also use your own flow cells, obviously. But we do provide custom-made flow cells as well.
I guess his follow-up question is it possible to do high throughput experiments in the C-trap setup, or is it always one molecule at a time?
Well, it's always one molecule at a time, but in terms of high throughput, in terms of like doing it in a more efficient way or doing it faster, as I mentioned, you can automate everything in the software. So it would be still one molecule at a time, or I don't know, depending on your experiment, you might have multiple DNA attached to the beads, but it would be one at a time, because you only have 2 traps, right? But in terms of how we define high throughput experiments in these kind of assays is based on how fast you can do collect data, and that is possible with automating the whole workflow with software.
Yeah. Does anyone have any other questions, or is it? Oh, okay. So Louis is asking, since VLPs were mentioned, to what extent is the system compatible with higher levels of biosafety, for example, viruses in biosafety level 2 labs or upwards? In other words, is the system completely closed, or are in order to clean up actions required?
Well, yeah, as I showed in the, maybe I can show the system again. Let me go back to, sorry. You can close the system, but at the same time, you do want to clean up as well if there is any spill on the system. I don't know if that was what they asked or not, but the ones that I showed you here, the examples, those are not actual, like they're not the virus particles, those are not actually viruses. They act like virus particles, so they're not dangerous or pathogenic. But in terms of how close the system is, I'm trying to, don't know why it doesn't exit the, okay, there we go. So I'll show you how the system looks again. I hope it's clear. So here you can see the lid here that can be completely closed. So, and you want that for other safety reasons in terms of like you working with IR lasers. And so as when you open the, there are all these safety features that when you open the lid, the IR laser shuts off. But you usually have the lid closed. And I don't know if that answers your questions or not.
I think it's also important to add that, that once you load in the samples, it's all on a closed system. So yeah, you just clean it and you reuse all the components. And I think there are also a few C traps that are in biosafety level 2 labs. So as long as you can get it in the lab, I think it should be fine. Shiba, he was saying that for his previous question, in terms of the throughput, compared to like a magnetic tweezer-based assay, I guess what's the comparison in terms of throughput?
Well, it depends even for magnetic tweezers. Well, in the setups that you would apply, you can apply forces to multiple beads at the same time. Yeah, obviously that would be higher throughput. But even for magnetic tweezers, there are systems that you would look at the single bead at each time, or you look at multiple beads at the same time. But in this case, because you only have 2 traps or maximum 4 traps, that is the limit. But we do have another technology called AFS that I didn't talk about today, but you can find information on the website where you would use acoustic forces and you would pull on many beads or many molecules at the same time. So if you're looking into higher throughput experiments, those are the way to go. But again, you won't have fluorescence imaging capability in those cases.
Okay, if there are no other questions, thank you again, Nasran, and thank you all for attending and listening to us. If you have any other follow-up questions, or if you want to learn more about the technology, just feel free to reach out to us. I think everything can be found on our website. Yeah, thank you guys. Thanks.
Yeah, thank you. Have a good day.